So our first speaker will be Shipei Xing. He's a postdoc at UCSD, and he's going to talk about structure annotation of full-scan MS data, a unified solution for LCMS and imaging MS analysis. And just a reminder for everyone, please be muted during the presentation. And we will have the question section after the presentation, and you can raise your hand. And also, if possible, enable your video while asking questions. And the question part will not be recorded. All right, Shipei Yao Tan.
Thanks. Yeah, I'm gonna quickly share my screen right now. Can everybody see it?
Yes.
Okay, nice. Thanks. Okay. Good morning, everyone. Yeah, I'm Shipei Xing. I'm currently a postdoc in Peter Darson Lab in UCSD, and today I'm gonna briefly talk about my first work during my postdoc. It's about structure annotation of full-scale MS data, a unified solution for LCMS and imaging MS analysis. Okay. So, yeah, the start of this project actually raised from the thought of reusing the enormous amount of public MS1 data. And the public metabolomics repositories are growing very fast. Up to now, we are approaching 15,000 datasets across main metabolomics repositories, including GMPS, Metabolomics Workbench, and Metaspace. Where the first three, they are mainly for LCMS metabolomics data, and Metaspace archives imaging MS data. And if you look at the data files in each repository, however, there are quite amount of MS data files that only have MS data. While we have been focusing on reusing all the public 10 MS spectra, and these full-scan MS data files, they have not been fully reused or reanalyzed sufficiently. And as a conservative estimate, these represents more than 40% of the total metabolomics data yet to be delved into. And it is generally known that during MS1 scans, many molecular ions may undergo in-source fragmentation, which generates fragment pieces that also appear in MS1 data. And these in-source fragment ions, they exhibit fragmentation patterns very similar to those observed in collision-induced dissociation MS2 spectra. And this provides the possibility of leveraging such in-source fragments as a handle to create pseudo-MS2 spectra or composite spectra. We can then easily perform, say, peak-to-peak correlations along the retention time domain to generate such pseudo-MS2 spectra, and then leverage them for metabolic annotation with ever-expanding reference MS2 libraries. So now that we can generate these pseudo-MS2 spectra, and how do we associate chemical structures with them? For a usual MS2 spectrum, we would match it against the reference library, and hopefully you get a nice matching score, which provides the structure annotation. And what about for pseudo-MS2 spectrum? So even if the true structure is in the reference library, as we have fragments of different ion forms that also correlate with the molecular ion, and sometimes we'd have Mr. cluster fragments appearing in the same pseudo-MS2 spectra as well. And these ions clearly diminish the spectrum matching results. And in such cases, how can we get a confident match? And from my perspective, I would say reverse spectrum match could be a perfect choice for annotating such a pseudo-MS2 spectrum. It works in the way that, say, when you match an experimental spectrum to a reference, you treat the reference spectrum as a template. And every non-matched peak in the experimental spectrum will thus be discarded during the scoring phase. Say, it is like if you see a dog with cat ears, you know that these ears, they do not belong to him according to your template dog image. So you will get a good match if you just ignore these ears. And here's a bit more information about reverse spectral search. It is actually used to be used for resolving chimeric spectrum. And my colleague, Vincent, he collected the data of drug standards with and without physics matrix, where you can see here are two example annotations of two drug molecules. where in the chemical standards, the spectra are pretty pure, and even homophysis, they have the chromatic spectra is pretty heavy. And then using the reverse spectral search, you get better cosine matches than the usual spectral search. So is such, I think reverse cosine or reverse spectral search could be a very good strategy to annotate pseudo-massive spectra. And we could also leverage reference spectra of different ion forms. Go back to LCMS data with big correlations with metabolome features along the detection time. We can generate these pseudo-massive spectra, and then they can be further annotated using MS libraries through a precursor-tolerant reverse spectral search. And here is a concrete example of annotating full-scan data in LCMS. We used the NIST human feces sample. We collected data under DDA modes and full scans with different in-source energies from 0, 10, and 20 volts. And you can see here is an example of annotating phenylalanine using its pseudo-MS spectra by matching against two reference MS library. And the in-source fragments, they exhibit very similar patterns as in reference MS libraries, and they show well-correlated peak profiles. And moreover, even we apply our approach on data-dependent data, where we have both MS1 and MS2 data, we obtained 40% more annotations using MS1 annotation strategy. And you can also see those low abundance metabolite features that can be also annotated. So you can see here is box plot of all the annotated features using different annotation strategies. Here is their the box plots of their peak heights of annotated features. And MS1-based annotation is indeed providing very similar annotations to those collected to those obtained from MS2 data. Also, in terms of annotations, their compound classes, MS1-based annotations, it does cover a broad diversity of compound classes, but there are clearly some classes they are underrepresented because some compound classes, they don't generate sufficient in-source fragments. And we further tested our strategy on the variety of sample types, including mouse species, micro-isolates, environmental DOM samples, and plant extracts. We obtained annotations in different compound classes as well. And specifically, here is an example of annotation in the public IBD dataset, where we were able to construct a molecular network using pseudo-MS2 spectra only. Well, for a common molecular network, these were constructed based on MS2 data, where here we use pseudo-MS data to construct such a molecular network, where you can see all the tyrosine related molecules. They are well clustered in the same group, and MS2 mirror plots allows us to annotate new isolipids using existing ones. And it is even more exciting that we can also generate such a pseudo-MS spectra from imaging data, where we correlate ion images in the spatial matter rather than in the retention time domain. And to give some background introduction of imaging data annotation, where in the year of 2016, Steerdorf Group, they published a FVR-controlled strategy for metabolite annotation in imaging data, where 3 scores were involved and integrated, including spatial chaos, spectral, and spatial isotope patterns. And these scores were integrated into a final score called MS1 score. And just to give you more information about spatial chaos, the measure of spatial chaos quantifies spatial informativeness within the image of the principal peak. And more recently, they developed a machine learning approach that also integrates absolute or relative NZ error together with the mentioned 3 measurements to achieve metabolite annotation. However, it should be noted that all these approaches, they are on the level 4 annotations, which means that all these annotations, they are essentially annotations of molecular formula. And then we would have to go to the chemical database, such as HMDB or lipid maps or Campbell or PubChem to retrieve structure candidates. And here is an example annotation on metaspace. You can see this is a mouse brain dataset, and then this is putative lipid molecule. And you can see design with very high MS score and very low FTR estimation of 5% FTR estimation. However, it has more than 50 candidate structures in the structure database, where you can see we have candidates of both PCs and PEs. And we cannot provide evidence for the chainless either. So we need more evidence to show which lipid it is PC or PE, and which chainless it is. So we are trying to go beyond molecular formula annotation to achieve level 2 or level 3 annotations. We're trying to give more insights into the structure. And how to do this? By leveraging the pseudo-MSU spectra. Say, in this case, we build pseudo-MSU spectra based on ion image correlations. And first, we conducted a standard spot-on experiment. This is a very nice collaboration with Mans and Theodore, and they spotted 7 chemical standards on the mouse liver tissue, including, you can see, ATP, ADP, adenosine, guanine, glutamic acid, glutamine, and glutathione. And eventually, except for guanine and glutamic acid, all the other 5 standards, they can be competently annotated. And these 2 cannot be annotated because they have very low MZ values, and then we do not observe their in-source fragments in the collected MZ range. Because for a common imaging experiment, people will start to collect MZ from 100 or even 150 or 200. And here are some other in-depth annotations of ATP, specifically for this molecule, where we observe other in-source fragments. Say we have a loss of a phosphate group or 2 phosphate groups, or with another water loss as well. So you can see with all these ion images correlated together, we were able to create such a pseudo-MSU spectra and then give us an annotation of ATP molecule. So this really gives us some more structural insights instead of just giving us a molecular formula. And going to the real imaging data sets, we were able to annotate lipid molecules in the mouse brain. And all these molecules have been verified using LC-MS data. Say we have light PC and PC lipids with different chain lengths and different ion bonds. We observed the characteristic PC hat groups 184, and they can be well correlated in the ion images correlations, so that we give more insights into that this is actually a PC, it is not a PE. And other than these molecules, we also we were able to annotate the amine molecule, and you can also see these clear, nice correlations observed between their ion images of the in-source fragments and then its molecular ion. And in a mouse body data set, we were able to annotate carnosine, and it is found to be localized in the mouse brain and mouse tissues. And this actually aligns with its dual role as a neuroprotector and a mouse performance enhancer. And similarly, this is a single cell data set of human hepatocytes. We were able to annotate diacylglycerols using its in-source fragments and pseudo-MSU spectrum. And yeah, here are the perspectives. As we all know, the LC-MS library is growing pretty fast. And it is so exciting to see that if we can leverage such resources to annotate imaging data as well. And on the other hand, currently, the GMPS ecosystem is able to perform molecular networking for LC-MS data using the collected MS2 spectra. And we anticipate this can be further extended to pseudo-MS2 spectrum, where LC-MS full scan data and imaging MS data will also benefit from such developments to discover new metabolites and unravel more biological insights. And Currently, this tool is already available as a Python package, and it's called MS1 ID, and it's also under development. So you're welcome to test it out and contact me if you have any suggestions. And I would like to acknowledge all my lab members and all the wonderful collaborations with Zero Lab and Minxun and Oliver as well. So yeah, that's all. Thank you. Any questions?